Photosystem 1 begins in the thylakoid membrane when antenna pigment molecules absorb photons of light and pass the energy to the chlorophyll 700 molecule in the reaction center of photosystem 1. This light energy causes electrons from the chlorophyll 700 molecule to become energized. The energized electrons leave the chlorophyll 700 and jump over to the electron transport system where they are picked up by a compound called photosynthesis. Photosynthesis transfers each electron to another electron acceptor. The electrons cascade from a high energy level to a lower energy level. The cascading of electrons down the electron acceptors is called the electron transport chain. The electron transport chain releases energy which will be used to move hydrogen ions into the thylakoid. When a proton gradient is established, the gradient will drive the synthesis of ATP. The electrons cascaded from chlorophyll 700 are not returned to chlorophyll 700. Instead, they are stored in NADPH. These two electrons and a proton from the breakup of water during photosystem 2 are picked up by NADP, forming NADPH. When chlorophyll 700 gives up its electrons, it becomes positively charged and cannot give up another electron. The 700 chlorophyll has what could be referred to as an electron hole. The needed electrons are going to come from photosystem 2. Antenna pigment molecules absorb photons of light and pass the energy to chlorophyll 680 molecule in the reaction center of photosystem 2. This boosts the electrons in the chlorophyll 680 molecule to a higher energy level. As in photosystem 1, the electrons cascade down an electron transport chain giving off energy. These electrons are then moved to photosystem 1, where the process can begin again. When chlorophyll 680 gives up its electrons, it becomes positively charged. This positively charged P680 molecule produces a strong pull on the electrons in water molecules. In a process known as photolysis, water is split. The water molecule breaks into protons, electrons, and oxygen. The energy from the electron transport chain is used to pump hydrogen ions to the inside of the thylakoid. This raises the concentration of hydrogen ions, and therefore the positive charge, inside the thylakoid. The remaining H+ ion from the water is picked up by the NADP, joining the two electrons that NADP received from photosystem 1, and NADPH is synthesized. The oxygen released from the water combines with another oxygen and is used either for cellular respiration or is released into the atmosphere. The electrons released from the photolysis of water pass to the chlorophyll 680 to replace the energized electrons transported to chlorophyll 700 along the electron transport chain. Since non-cyclic photophosphorylation is a constant and ongoing process, light and NADP are constantly variable, and photosystems 1 and 2 reoccur almost instantly. Once there is a high concentration of H+ ions in the thylakoid, chemosis takes place.